What if one of the greatest medical breakthroughs in history happened because of a messy lab? In 1928, a brilliant but famously disorganized scientist named Alexander Fleming made an astonishing observation. He left his petri dishes exposed, and what he found growing on 1 day later changed medicine forever. This is the incredible true story of penicillin, the life-saving antibiotic that emerged from an overlooked fungus and a simple oversight. Discover how a small mistake launched the age of modern medicine. In September 1928, Alexander Fleming, a Scottish physician and microbiologist, returned to his laboratory at St. Mary's Hospital in London. He had been conducting research on staphylococci, a common type of bacteria responsible for various infections. His lab, often described as somewhat untidy, contained numerous petri dishes with bacterial cultures. Upon inspecting his old cultures, Fleming noticed something unusual on one particular petri dish. This dish, which had been left uncovered for some time, was contaminated with a distinct mold. Crucially, around the mold colony, there was a clear, bacteria-free zone where the staphylococci had failed to grow. Fleming immediately recognized that this clear zone indicated the mold was producing a substance capable of inhibiting bacterial growth. He carefully isolated the mold and began to conduct further experiments. This observation marked the accidental inception of a groundbreaking medical discovery. Further investigation identified the contaminating mold as a species of Penicillium, specifically later confirmed to be Penicillium notatum. Fleming found that the mold's broth culture, when filtered, contained an active substance that possessed strong antibacterial properties against many common pathogens. His tests demonstrated that this substance could kill or halt the growth of a wide range of harmful bacteria, including streptococci, meningococci, and diphtheria bacilli. However, he noted it was not effective against all bacteria, such as the typhoid bacillus. This selective action was a significant early finding. In 1929, Fleming published his findings in the British Journal of Experimental Pathology, naming the active antibacterial agent penicillin. He described its inhibitory effects on various microbes and suggested its potential as a topical antiseptic. However, he faced challenges in isolating and purifying the compound in stable form. Despite his significant discovery, Fleming initially struggled to extract a stable and concentrated form of penicillin. The substance he produced was highly unstable and difficult to mass-produce, limiting its immediate application as a therapeutic drug. Consequently, his work did not gain widespread attention for over a decade. It was not until the early 1940s that a team of scientists at the University of Oxford, led by Howard Florey and Ernst Chain, revisited Fleming's work. They successfully developed methods for purifying and mass-producing penicillin. Their research involved extensive chemical and biological experimentation to stabilize the compound. The purified penicillin proved to be incredibly effective in treating bacterial infections in humans, and its development accelerated during World War II. It rapidly became a critical medical resource, saving countless lives on battlefields and in civilian hospitals by combating infections like pneumonia, gangrene, and sepsis. Penicillin's widespread use marked the dawn of the antibiotic era, fundamentally transforming modern medicine's ability to combat infectious diseases. This profound impact earned Alexander Fleming, Howard Florey, and Ernst Chain the Nobel Prize in Physiology or Medicine in 1945 for their pioneering work. Thanks for watching. Please subscribe to our channel.